This video provides a critical appraisal of nicotinamide riboside, or NR, and nicotinamide mononucleotide, or NMN, evidence for longevity and anti-aging. We explore the NAD plus translational gap and the evidence quality, bias risks, and clinical reality. Short-duration trials consistently show that NR and NMN dose-dependently increase circulating NAD-related measures in the blood. However, anti-aging efficacy, which requires translation to tissue-level biology and functional lifespan improvement, remains systematically inconsistent. The human evidence base is dominated by surrogate biomarkers. The translational funnel illustrates the progression from preclinical models to clinical aging outcomes. Challenges arise in short-term randomized controlled trials due to assay and compartment specificity and dose and duration heterogeneity, yielding highly heterogeneous results. Biomarker shifts for both NR and NMN are often seen in the blood, but the magnitude is variable and sometimes assay or compartment dependent. Metabolic and functional outcomes show mixed or null results, and cognitive outcomes have limited or no improvement. For NR, the translational disconnect is evident. In the null clamp, 12 weeks of high-dose NR in obese, insulin-resistant men showed zero improvement in insulin sensitivity. The muscular disconnect showed increased muscle NAD metabolome, but no mitochondrial bioenergetics improvement. The functional caveat showed modest 6-minute walk improvement in PAD patients, relying on a lenient statistical threshold. NMN studies show extreme heterogeneity. A positive signal in pre-diabetic postmenopausal women showed increased muscle insulin sensitivity, but muscle NAD content was unchanged. Null or mixed signals were found in older men with diabetes, showing no improvement in grip strength or walking speed. The overall evidence is complicated by reliance on proprietary metrics. The compartment disconnect highlights the difference between serum or whole blood and striated skeletal muscle. Blood is highly responsive to supplementation, but trials show that blood NAD movement does not guarantee tissue restoration, where target engagement is inconsistent or unmeasured. The time match illusion illustrates the gap between the short duration of current randomized controlled trials, which last only weeks to months, and the clinical arc of human biological aging, which takes decades. Short trials cannot validate claims about slowing biological aging. Methodological failure modes include multiplicity and fishing, where multiple endpoints are exploited without statistical correction, and selective exclusions, which can introduce severe biases. Crossover missingness involves poor handling of trial effects. And quote, sample size constraints mean trials are often underpowered for modest functional differences. The statistical illusion of overstating functional benefit often results from pre-specifying a highly lenient one-sided p-value threshold, such as less than 0.10. This artificially inflates the appearance of clinical benefit, especially in small sample sizes. Structural bias in the NAD plus evidence ecosystem stems from direct employment of authors by sponsors and intellectual property licensing involving inventors holding IP equity. This raises the probability of selective reporting, optimized endpoint choices, and publication bias. Short-term safety is generally established, as the substances are well tolerated at marketed doses with no severe adverse events. However, long-term safety remains unknown, with concerns about homocysteine rises, geriatric vulnerabilities, and interactions with cancer risk groups and oncologic therapies. Regulatory status varies by region. In the US, NR is a stable new dietary ingredient, while NMN status is contested. The EU has novel food authorization for NR chloride, but NMN faces scrutiny. Canada licenses NMN as a natural health product. Allowed to sell is distinct from, quote, proven anti-aging efficacy. The Grade Certainty Scorecard synthesizes the evidence. Certainty is moderate to high for increased circulating NAD, but low to very low for all other outcomes, including tissue NAD increase, metabolic and functional improvements, cognitive outcomes, and clinical aging outcomes. Future research requires pragmatic randomized controlled trials lasting 12 to 24 months in high-risk older adults. Methods must be standardized, independence must be enforced through large-scale publicly funded trials, and safety metrics must prospectively track geriatric endpoints. When consulting with patients, advise that these compounds move biomarkers but lack high-certainty evidence for slowing aging.